Thank you all very much for joining us today. It's a pleasure to speak with you. And as those of you in the audience who know me, this is a subject that is very dear to my heart in terms of predicting metabolism, going all the way back to my PhD days, back when I had hair. So what we're going to do today is, first of all, just tell you something that I'm sure most of you in the audience will already be very familiar with. We'll just talk a little bit about the role of DMPK in drug discovery. And then particularly focus on the impacts of drug metabolism. And then we'll talk about some of the challenges we have in drug discovery in relating to drug metabolism. First of all, focusing on cytochrome P450s, then looking beyond P450s at the impact of other phase 1 and phase 2 drug metabolizing enzymes. And then we'll actually go beyond looking at human metabolism and look at metabolism by preclinical species. And along the way, what we'll be doing is looking at how these in silico methods can really help us to address some of these challenges. And as Catherine says at the end, there'll hopefully be plenty of time for Q&A. We'll be really happy to answer your questions. And anything that we don't have time to answer live today, we'll certainly follow up afterwards by email. So first of all, the roles of DMPK and DMPK scientists in drug discovery. First of all, and we all very much hope we can do this, is identify those high-quality compounds that have appropriate ADME properties and pharmacokinetics for the objectives of our project. Of course, we don't always find those, at least not initially, and so another thing we want to do is to highlight potential liabilities in our compounds that need to be addressed in lead optimization. So providing that guidance to the medicinal chemists on how they should focus on optimizing the compounds to achieve that ideal balance of properties. Moving downstream, once we're beginning to think about nominating candidates for preclinical studies, also making sure we're giving some guidance on the appropriate species for preclinical PK and toxicity studies, ensuring particularly appropriate exposure and coverage of metabolites. And of course, it's also about designing the appropriate experiments to achieve those three key goals, and also manage costs and reduce the use of animal studies along the way. Now, in this webinar, we're really going to focus on the questions relating to drug metabolism and highlight some of the ways that in silico methods can really help to achieve these goals. And just to be clear, today we're really talking about the application of in silico models. If you want to know more information about the underlying methods and their validation, for example, we've referenced numerous publications in our slides, and also there are other webinars that you can watch, for example, the one with a link here at the bottom. And as Catherine says, all of these are available on our website if you just want to drop in and watch those and find out a bit more about what's going on under the hood. Now, if we focus specifically on drug metabolism, of course, the role of metabolism in the body is the elimination of both endo and xenobiotic compounds. And of course, we have evolved numerous pathways for elimination, many different enzyme families and isoforms. But ultimately, the goal of those processes is to increase the solubility of compounds, which will then lead to the excretion of those metabolites from the body. But these metabolic processes create a number of challenges we need to overcome in drug discovery. So first of all, and very commonly, we find compounds that are rapidly metabolized. This can lead to high clearance or low oral bioavailability, for example, and we need to resolve that to achieve appropriate exposure. We also sometimes find the formation of reactive or potentially toxic metabolites. Of course, that's pretty bad. We want to avoid that wherever possible to avoid any toxicity, safety issues, or side effects. We see genetic polymorphisms in the expression of these enzymes, and that can lead to significant impacts on exposure in different patient populations. And there is the potential for drug-drug interactions. If we co-administer multiple drugs, then, for example, inhibition or induction of enzymes by one drug can lead to variability in the exposure of another, again, leading to potential safety issues and side effects. So these are some of the challenges that we have to overcome when we're optimizing compounds in drug discovery and selecting clinical candidates. So now we're going to drill down on each of those in a bit more detail. And first of all, we're going to focus on cytochrome P450s. Why do we want to focus on P450s? Well, it's where we usually start in these subjects. The P450s are, of course, a ubiquitous superfamily of heme-containing monoxygenases, and they are responsible for roughly 70 to 80% of observed drug metabolism. This can lead to, again, rapid clearance or low bioavailability, drug-drug interactions. There are significant P450 polymorphisms that we need to take into consideration. And again, P450s are certainly responsible for bioactivation to form reactive and toxic metabolites. And it's also important to note, as illustrated here, that there are numerous different isoforms of P450 that are responsible for drug metabolism, led by the usual suspects such as 3A4, 2D6, 2C9, 1A2, and on down the list here. So it's not a simple enzyme family to deal with by any means. Now, thinking about the first problem that we just mentioned there in terms of metabolic instability, which can lead to high clearance or low oral bioavailability, typically we would assess this in microsomes. And so typically we might look at the intrinsic clearance, half-life, or indeed turnover in human liver microsomes, which would, high in turnover clearance or short half-life, typically would indicate rapid metabolism by cytochrome P450s. And if we observe this, we can use in silico models to help to guide that optimization of compounds to improve their metabolic stability. So, for example, in silico models can identify the sites of metabolism, otherwise frequently known as metabolic hotspots, and give us guidance on where we need to block that metabolism to improve stability. And if we look here on the right-hand side, we can see the predicted regioselectivity of metabolism by the major isoforms are responsible for metabolism of this compound. And you can see that it's likely to be hydroxylation on these different methyl groups will be the predominant sites of metabolism. But that in itself is important information, but not sufficient to really guide the optimization of these compounds. An additional piece of information we can provide is what's known as the site lability. This is an absolute measure of reactivity to P450 metabolism. So we look at this in terms of this metabolic landscape, where the different sites are sort of ranked in terms of their lability or the vulnerability of those sites to metabolism. And we can sort of summarize this in a value called the composite site lability, which is a measure of the efficiency of product formation and hence an increased risk of rapid clearance. And so actually there's more information. There is actually a third highly labile site here on this compound, which although it's responsible for a small proportion of metabolism, in absolute terms, it is also highly labile. So if, for example, we were to modify or block these predominant sites, what we're likely to see here is metabolic switching to an also highly labile site and hence see a relatively small decrease in the overall rate of metabolism, i.e. we have more work to do on this compound. Now, it's important to note that this site lability, or composite site lability, is not a prediction of the overall rate. It is one very important factor, but other factors are important. For example, and not least binding affinity. If a compound doesn't bind to a P450, it doesn't matter how labile the sites are. And we'll come back to that question in just a moment. But first, I thought it would be interesting to look at a specific example of this to show how one can use this in practice. And I'm just going to show a very brief and simplified version of a study that was published actually using much older versions of the models that we used back in 2004, and it was published in Bioorganic Medicinal Chemistry Letters. But this focused around the design of analogs of bucarone. So bucarone is a drug. It's an anti-anxiolytic and a ligand for 5-HT1A. It's got excellent receptor affinity, 25 nanomolar, but it does suffer from poor and variable oral bioavailability, and that is driven by metabolism by cytochrome P450, specifically CYP3A4, and one can see this in vitro. It has a half-life in vitro of 4.6 minutes. And so the goal of the project was to identify analogs of bucarone with significantly improved CYP3A4 stability, in this case aiming for greater than threefold improvement, so roughly 15 minutes, but also, of course, retaining receptor affinity, and the project was willing to sacrifice about an order of magnitude. And in terms of the experimental data that's available, here we're highlighting the primary sites of metabolism that were identified experimentally. So the para position on the pyrimidine, the alpha position to the nitrogen in the piperazine, and also this spirocyclopentane here have been observed experimentally. And so let's actually do this in practice just to show you what this looks like. So I'm just going to jump in this case, into our StarDrop software. And here you can see I just have bucarone and a couple of analogs. We can easily calculate the predicted metabolism by cytochrome P450s. So let me just do that. This actually appears instantly, and just to be completely transparent, these are pre-calculated results. They're stored on the server when you calculate them to save time and not reproducing the same results multiple times. In practice, that might take a few minutes to perform that calculation. But this here is bucarone itself, and just really to show you here, this is the predicted regioselectivity of metabolism, in this case by 3A4. We'll talk later about which enzyme in which P450 model, so I'm just going to hide those away for the moment to focus on the metabolic landscape and the sites of metabolism. But what you can see here, again, rather nice to see, is those predominant sites of metabolism on the pyrimidine and adjacent to the nitrogen on the piperazine are indeed predicted to be the major sites of metabolism. And although it's a minor site, you can also see here on the spirocyclopentane. So, this is giving us a good representation of what's happening experimentally. And so what we can see here, there are two highly labile sites, again, on the pyrimidine and at that alpha position there. Those are the sites we would want to focus on, but maybe also looking at this end of the molecule here. And what you can see are two other analogs that were designed to address some of these questions. We can, again, select those and calculate their metabolism by cytochrome P450s. Again, seeing the results. We can see here significant improvements in that metabolic landscape. There are no highly labile sites. Here you can see we've blocked that para position here and added some steric hindrance there. And if we look at the the second example here, you can see actually making modifications again with a fluoro, blocking that site, and at the changing that spirocyclopentane into a gem dimethyl, also significantly reducing that metabolic landscape. And that is also reflected in this composite site lability that you can see summarized here. So driving that composite site lability down has a significant effect. And when these were tested experimentally, you can see indeed, in this case, a half-life of almost 80 minutes. And here again, achieving that goal of about 15 minutes while retaining good activity against the 5-HT receptor. So you can see how we can find these sites, provide that guidance to blocking those sites to improve their metabolic stability. Just change it back. There we go. And so just to summarize those results here, you can see removing those highly labile sites, achieving a significantly lower composite site lability, indeed increases that metabolic stability. Here again, you can see a couple of other examples where we can significantly increase that half-life in vitro. So the composite site lability is one factor we can influence to improve the metabolic stability, but I mentioned before that it is not itself a prediction of the rate of metabolism. Why is this? Well, the P450 catalytic cycle is very complex and actually has many potentially rate-limiting steps. So what this means is in practice that quantitative prediction of metabolic rate, whether that be intrinsic clearance or half-life, for a diverse chemistry, for a global data set is intractable based on structure alone. So we have to consider, for example, binding. The KM in Michaelis-Menten. Compound doesn't bind, it doesn't matter how labile it is, it won't be rapidly metabolized. The reduction steps can be rate limiting, particularly influenced by type 1 and type 2 binding. And the decoupling pathways can also reduce that rate of metabolism. significantly. Whereas the composite site lability is looking at one very important factor, which is the efficiency of this product formation step. However, what's good news is that it is certainly possible to build local models of metabolic rate. That's because within a given chemical series or project's chemistry, it's likely that only a small number, one or two of those factors, will be very influential on the overall rate of metabolism. So let's look at an example of this. This was an example where we used the auto modeler in our StarDrop software to build a model of the rate of metabolism by CYP3A4. This was expressed as a log of the rate constant. The model was built based on a 100 compound data set from a project. The descriptors we used were indeed that composite site lability that captures that efficiency of metabolism, but also two factors that are very important for determining the binding efficiency, binding affinity, the log P, and the polarity of the compound. In this case, we're looking at the log of the neutral fraction at physiological pH. And what you can see here for the independent validation and test sets is a really very good correlation with a coefficient of determination of almost 0.7. And just looking at the specificity of the model, particularly what you can see is of those compounds that were predicted to meet the stability criteria, the large majority of those were found to be stable in practice in those independent test sets. So this was then applied to a virtual library of 13,000 compounds designed by the chemists in this project. From that 13,000, 40 compounds were selected for predicted stability and potency. And from these 40 compounds, 4 new potent and stable series were identified, so illustrated here. All 4 of these series, A, B, C, and D, are identified to have highly active compounds and to meet the metabolic stability criteria, in this case, for this project, of greater than 20-minute half-life in vitro. So within these local contexts, one can make highly quantitative and accurate predictions of the rate of metabolism. But metabolic rate is not the only thing we're concerned with. We mentioned genetic polymorphisms, and certainly the polymorphisms in some P450 isoforms, such as 2D6, 2C9, and 2C19, are quite prevalent. This leads to poor metabolizer phenotypes and substantial subpopulations, particularly within certain ethnic groups. And that phenotype can lead to significant increases in exposure to the drugs in those poor metabolizers. And so this is something we would want to identify early on, the potential for that. And what we have here is what we call our Witch P450 model. So this identifies the specific isoforms that are most likely to be responsible for metabolism of a compound. You can see it's sort of represented here as a sort of pie chart. This is looking at the relative likelihood of metabolism by different isoforms. Again, it's not a prediction of the relative rate. We've talked about predicting rate a moment ago. So this is looking at how likely a given compound is to be metabolized by each of these isoforms. And in examples like the one you can see on the right-hand side, which is metoprolol, if you see a prediction that the metabolism is predominantly by a single polymorphic isoform, this would indicate an increased risk of exhibiting differences in exposure due to the polymorphisms in this case, CYP2D6. Now, what one would want to do in response to a prediction like this, of course, is to experimentally verify this. So we might use something like a P450 phenotyping experiment using recombinant enzymes to really dissect which of those isoforms are likely to be responsible for clearance of that compound. And also we can think about the risk of formation of reactive or toxic metabolites. So we know that metabolism by P450s can lead to the formation of these metabolites. We might observe experimentally, for example, time-dependent inhibition, which is caused by irreversible binding to the P450 enzyme. Or, for example, following metabolic activation, you might see formation of glutathione conjugates. Now, to be clear, we cannot yet directly predict the reactivity of metabolites. I'd say watch this space. It's some active research that we're working on. However, what we can do is look for patterns of metabolism that correlate with the formation of reactive metabolites. In this particular example, we'll look at a series of PDE10A inhibitors published by Chino et al. A few years ago. And this is an example of one of the compounds from the series. You can see here that in the paper that they suggested that the metabolism at the 7 position on the imidazopyridinone ring may be responsible for formation of that reactive metabolite. But what's interesting, if we actually look at the predictions for this compound and a number of others within that series, here you can see the predictions is really that the vast majority of metabolism is likely to occur due to O-demethylation or N-demethylation here. And in fact, there's no predicted metabolite, no significant predicted metabolism at that 7 position. On the other hand, what we can see here is a strong correlation between N-dealkylation on that same ring system here, and the experimentally observed time-dependent inhibition. This is shown here as the percentage activity remaining after a 30-minute incubation. And so a low value here indicates a high degree of time-dependent inhibition. And so this correlation, admittedly, it's only 3 data points here, but there's a very strong correlation between the degree of N-demethylation and this time-dependent inhibition, which would suggest that reducing N-dealkylation is one approach to improving the risk profile of these compounds. It's likely that the N-demethylation there leads to rearrangement and activation of that ring system. Okay, so those are a few examples related to P450 metabolism, but that's not sufficient. We really want to go beyond P450s. Why is that important? Well, as we've just heard earlier, 20 to 30% of the direct metabolism of drug molecules is due to non-P450 enzymes. Here you can see a nice analysis of the top 200 drugs and looking at the proportion of metabolism the proportion of different enzyme systems responsible for metabolism that are not P450s. For example, things like UDP glucuronol transferase and sulfotransferases, for example. And of course, when we think about phase 2 metabolism, so after that modification phase by often P450s, we'll then see, for example, conjugation by UGTs, sulfotransferases, and others. And these are the products of that metabolism are also very important. And finally, as chemists have become much more adept at avoiding P450 liabilities, what is being observed, at least anecdotally, is that metabolism by these other enzymes are becoming more and more important routes of clearance that we need to consider. So enzymes such as aldehyde oxidase and flavin-containing monoxygenases. So we need to think about that in terms of our compounds. If we focus purely on microsomal stability, this can lead to unexpected metabolism by other enzymes in late-stage studies. To help us to think about that, we have also developed what we call the which enzyme model. This identifies the enzyme families that are most likely to metabolize a compound, by analogy with the which P450. So again, represented by these pie charts here, you can see the relative likelihood of metabolism of a compound by each enzyme family. And this can really help to prioritize appropriate in vitro and in vivo PK studies. A couple of actual examples of this are 2 clinical candidates, felnitamol and cabazitaran. These were actually candidates that failed in the clinic due to poor oral bioavailability and exposure, despite the fact that they had passed all of the usual preclinical PK studies. So let's just look at the prediction of metabolism by these compounds. I'm just going to jump back into the software. I'm just going to bring up these 2 molecules here. And again, I'm going to predict the metabolism of these compounds. Again, the results appear instantly just for convenience because I have pre-calculated those. And what you can see here in terms of the which enzyme and which P450, you can actually see summarized in the table as well that these are predicted to be largely metabolized by aldehyde oxidase, not by P450s at all. So here you can see phenylnidamol, here you can see carbazeran, again, predominantly aldehyde oxidase. And indeed, we can also predict the sites of metabolism. So here you can see for phenylnidamol, there are actually 2 sites of metabolism that are predicted for aldehyde oxidase. And we can even drill down a bit further and look at the details, and you can see the most likely site here, the statistically most likely site is this position here, which has a slightly higher probability than this position. So we can, again, provide that guidance as to where the compound can be modified to improve that metabolic instability. So in this case, these predictions that aldehyde oxidase is a very likely candidate for metabolism of these compounds would lead to choose some different experiments to do earlier on in drug discovery, not wait till the clinic, but for example, looking at PK studies in guinea pig or rhesus monkeys. They have good aldehyde oxidase activity, whereas rats and dogs have little or no aldehyde oxidase activity. So this would help to prioritize those in vivo experiments, or indeed look in vitro at hepatocytes, for example. Another thing we spoke about was drug-drug interactions and how we can reduce that risk. So, for example, in this case, we're looking at metabolism. One source of the risk of drug-drug interactions is if a compound is predominantly metabolized by a single isoform of a single enzyme, because inhibition or induction of that specific isoform by a co-administered drug can lead to significant changes in exposure. And again, we can look at that combination of the which enzyme and which P450 models to identify likely candidates for this case. So here, for example, for simvastatin, you can see this is predominantly predicted to be metabolized by P450s, and of the P450 isoforms, here we can see almost exclusively by CYP3A4. And again, there's a well-known drug-drug interaction between simvastatin and antifungals such as fluconazole and ketoconazole, which are potent CYP3A4 inhibitors. So we would need to be aware of that in the optimization of our compounds, or at least as we were moving forward into the clinic. And then we want to bring this all together and say, well, what metabolites are most likely to be generated by my compounds in vivo? Now, this is a really big challenge for in silico methods. If we generate all possible metabolites of a compound, which we can do, this can lead to an overwhelming number of predicted metabolites when we're considering multiple generations. So here I'm showing for dextromethorphan just 2 generations. Here's the drug itself. Here are its direct potential metabolites. And then there's the metabolites of the metabolites. That's 2 generations. And of course, there can be multiple generations formed in vivo. But even if we're just looking at 2 generations, we can see here the green compounds highlighted here are those that are observed experimentally, and they're a tiny proportion of the potential metabolites. And this is a well-known problem for knowledge-based methods for metabolism prediction. We can generate lots and lots of potential metabolites. We can find all the potential metabolites, essentially. But which ones are actually going to be observed experimentally that we need to be concerned about? We actually did an analysis of this for 92 compounds, again, published here in the publication at the bottom. And what we considered all the potential metabolites that could be generated for these enzymes that we are considering here. We actually did correctly identify 96% of the experimentally reported metabolites. In statistical terms, this is 96% sensitivity. But of those generated, only 4% of those were actually reported experimentally. Again, in statistical terms, this is a 4% precision, or another way we can think about it is we're predicting 25 times more metabolites on average than are actually observed experimentally. In fact, it's even worse here for dextromethorphan on the right-hand side. Yes, the sensitivity is 100%. We find all of the experimentally observed metabolites, but the precision is actually 2%. we're generating 50 times more metabolites than are actually observed experimentally. So we can do better than that by using this sort of mechanistic approach to predicting the most likely metabolites, the most likely enzymes and isoforms. Again, let me just quickly demonstrate that to you so you can see what it looks like. I'm going to go back into the software. I'm just going to look at this example for dextromethorphan here that we just saw previously. Going to select the compound here, and I'm actually going to ask it to generate the metabolic pathway for this compound. So this is looking at 2 generations. And again, that will appear very, very quickly. This is actually a very long calculation in practice because it's actually generating many different metabolites, and then for each of those metabolites predicting their metabolites. So this can, in practice, take hours. But again, for convenience, once you've calculated it once, it returns instantly. And what you can then see is, if I just click here, it actually generates a sort of metabolic pathway for dextromethorphan. You can see here a much smaller number of metabolites that are predicted as to be likely to be formed, giving us a much smaller pool to go look at to confirm experimentally. And if I just go back to show you those results in a little bit more detail, what you can see here on the right-hand side is, again, the same color coding. This is dextromethorphan, the drug. Here are the experimentally observed metabolites that are correctly predicted. And you can see that a much greater proportion of those predicted are actually observed experimentally. The precision is increased from 2% to 40% in this particular case. Now, it will vary from compound to compound. The larger, more complex the compound, the harder it is to achieve these very high precisions. But here you can still see in our independent tests a significant improvement in precision over the sort of boil the ocean and generate all possible metabolites. So this can really help to guide metabolite-ID experiments to focus on the most likely metabolites, particularly if we need to get into sort of resynthesis of compounds for quantification. So we've looked at a variety of ways we can predict metabolism by human isoforms and enzymes, but quite rightly pointed out that the first place we dose our compounds is not straight into humans, but into animal models. And so we might like to compare that human metabolism with the metabolism by various preclinical species. One reason this is very important is that choosing the most appropriate species for preclinical studies is absolutely critical. We need to ensure coverage of human metabolites in our animal models. And if they're not generated in our animal models, we do need to identify those and potentially synthesize those for further safety testing. And to help with this, one can make predictions for non-human P450 metabolism. In our case, we have produced models of mouse, rat, and dog. And we can use these to identify the most appropriate species to generate similar metabolite profiles and hence prioritize that experimental effort. And just to sort of show you a quick example of that as well, let's look at an example of sildenafil. So here you can see we can make a prediction. Again, I'll just focus on P450 metabolism, for which we have these preclinical models. So here again, you can see the summary we saw before of the human metabolism. But if we want to compare and contrast, what you can see here side by side are the human predictions and the predictions by the preclinicals for the preclinical species. In the case of the preclinical species, we don't have isoform by isoform data, so we have a model predicting for the species as a whole. And that means they're also slightly more qualitative. Instead of producing an actual quantitative regioselectivity, we're identifying primary, shown here in bold with the primary symbol, secondary and tertiary metabolites. Now, experimentally, in humans, the predominant site of metabolism is indeed this N-demethylation position here on the pyrazine, but there's also metabolism observed here, another N-demethylation site here as well. And so if we wanted to compare between the human and the preclinical species, really it's the mouse that is most likely to give us the coverage of the same metabolites that we're observing in our human experiments. So again, sort of summarizing the results that we just saw, the predominant sites are here, identified by the orange circle. And again, that is in excellent agreement with the expected or the observed, experimentally observed metabolism in mouse, which is published by Lee et al. Here. So it's just confirming that prediction is correct. So just to draw a few conclusions before we wrap up and answer any questions. Just to summarize, we've covered a lot of different models in these discussions of the different applications. So just to provide a summary of the models provided by our solutions. So we, for our P450 metabolism models, we cover the 7 major drug metabolizing isoforms of P450s in humans, as well as predictions of rat, mouse, and dog. And we predict both this metabolic landscape and the composite site lability that provides a significant additional information that we can use to guide the optimization of compounds. We also predict aldehyde oxidase and 2 different isoforms of flavin-containing monoxidase for phase 1 metabolism. And for phase 2 metabolism, we predict a range of different UGT isoforms as well as sulfotransferase. We discussed the witch enzyme and the witch P450 models that help us to identify which of those predictions is most relevant for our compounds and enables us to combine all of these different predictions to predict the metabolite profiles, and of course identify these issues regarding drug-drug interactions and impact of genetic polymorphisms. And we predict the metabolic pathways that we just saw as well. So we've covered a lot of questions here. We've looked at when we have metabolic instability problems, we can use the site of metabolism models to guide that design. We've looked at predicting the rate of metabolism. So this is a cautious yes for local chemistry. And we showed an example of QSAR models using the composite site lability to select stable compounds. We looked at the witch P450 model and way we can use that to identify potential risks around genetic polymorphisms. We use the site of metabolism models to look at patterns that of metabolism that lead to the formation of reactive metabolites. We use the witch enzyme model to ask questions around what enzymes from other families are likely to metabolize a compound other than P450s. And we looked at the combination of which enzyme and which P450 in terms of reducing the risk of drug-drug interactions. We combined all of those to look at metabolite profile predictions. And then we looked at the application of preclinical P450 models to identify the species most likely to provide good coverage of human metabolites. I do need to give acknowledge a lot of people. I have the pleasure of talking about all this great science, but of course it's the work of many people over many years, many of my colleagues at Optibrium, of course, but you know, this has been the culmination of really 2 decades of different work with collaborators and other mentors and other companies as well. And so really just sort of listed some of those sort of key people we've worked with over the years to help us to think about what are the key challenges to address and think about different modeling methods that can help to address those. And finally, as Catherine said, there are actually 2 ways now to access our metabolism models. On the left-hand side, you can see Symetra. Symetra was just announced a few weeks ago and is our new dedicated application for metabolism prediction. It's a cloud-based platform, and the same methods are also available as a plugin module to our Stardrop software, which is a complete package for small molecule design, optimization, and data analysis. Both of these applications have the same intuitive interfaces. I just gave you a quick example of how we can quickly run these calculations and visualize and interact with those results. They're exactly the same in both applications. And of course, within Stardrop, this is coupled with a very broad range of capabilities for molecular design, optimization, and data analysis, in silico modeling using other methods, generative chemistry, and 3D design, where again, Symetia is a dedicated platform for DMPK. So, thank you very much for joining us. I'm going to hand over to Catherine, who is going to talk us through the Q&A session. Thank you very much.
Thanks so much, Matt. We've got lots of questions that are already coming in. If you've got questions but you haven't asked them yet, then please do click that Q&A button on your screens and we will try and answer as many as possible. So let's start things off at the top then. We've had someone who has asked for a little bit more clarity about the difference between site lability and site of metabolism. I'm assuming this is to do with those reduced selectivity maps. Matt, do you want to take it away?
Yeah, absolutely. That's a great question, and it is somewhat hard to explain. I'm sorry I didn't explain that correctly. So when you're predicting the regioselectivity of metabolism, you're looking at the relative rates of these different sites. So these particular sites in this compound here are rapidly metabolized compared with this position down here. That's looking at, relatively speaking, how rapidly the product will be formed at those sites. But actually, you can think about this in absolute terms. So here you can see this third site is less labile, but that small change in lability actually translates into quite a significant change in rate. It's a non-linear relationship. And so this is why looking at the lability on an absolute scale is also very, very important. To go into a little bit more detail, this lability is site lability is calculated relative to one of the decoupling pathways, which provides a sort of a baseline rate against which you can assess the efficiency of that product formation. And so you're just basically comparing here sites with each other versus each site with this constant common baseline that they have of that decoupling pathway. I hope that helps. If not, I'll be happy to point you at our publications that really actually spell out the equations that describe that.
Well, thanks very much, Matt. On to our next question. Do our predictions take into account 3D structure? So, for example, is it possible to identify differences between enantiomers?
Yes, that's another really good question. And the answer is somewhat, but not extensively. So again, I've got a nice slide that will hopefully illustrate that, one of my backup slides here. So the way that we actually make the predictions is a combination of two different factors. One is actually the reactivity of those sites to the different reactions leading to metabolism by the different families of enzymes. So here on the right-hand side, you can see this nice little animation of the flavin-containing monoxygenase reaction mechanism. So those are, of course, performed using the 3D structures, and that would be sensitive to the different stereoisomers or enantiomers. The other factor we take into account is the accessibility of those sites to capture the effects of orientation in the binding site and steric hindrance from the substrate itself. These are actually captured by 2D descriptors that capture distances between potential sites of metabolism, like this para position here, and in this case, this basic nitrogen, sort of illustrated by this orange line. That is a 2D descriptor, so it doesn't capture the effects of stereochemistry. So typically, the models do not resolve very well between the different stereoisomers, and that is a limitation. Part of the reason for that is there's actually fairly limited data available for different stereoisomers, individual stereoisomers, where we have good sort of metabolism data, so that we can actually train models to capture those really quite subtle effects of stereochemistry. So yeah, it's not well captured by these models, that subtle effect.
Thanks, Matt. The next question is whether our which enzyme model will work for peptides with between 5 amino acids to 15 amino acids, or just peptides in general.
Yeah, and another really good question. And the answer, to be absolutely honest, is no, I wouldn't expect it to work well. Those models are trained on small molecule drug-like compounds. And so no, they're not trained for peptides. So yeah, unfortunately not. It might well be possible to train such a model. We are looking at models of peptides and peptidic macrocycles. As I'm sure you know, they tend to be much harder to make predictions for, partially because there's less data available, and secondly, because the enormous flexibility of those structures makes it really hard to know how best to characterize them and what types of descriptors to use.
Great, thanks, Matt. Looking at our list of questions, I think the next question will be, is it possible to teach the predictive models by uploading our own metabolism data?
Yes and no. It is certainly possible to retrain the models based on in-house data. There we really are using site of metabolism data. So if you have that sort of regioselectivity data, that is absolutely ideal. It is not possible for you to do that yourself yet. But it is something that our team are well capable of doing, and it's something we're delighted to collaborate with our customers to build these sort of tailored models on a sort of a customer by customer basis. And just to be clear, we're not talking about building that in and sharing it with other customers, but actually just doing a tailored model for your use.
Okay. So we've got a question here about how many parent compounds are used in the predictive models. I'm assuming that might be the auto model.
Absolutely. Yeah. So in the training sets for these models, the site of metabolism and the which enzyme, which P450 models, for each enzyme or for which isoform, we're typically looking at hundreds of molecules. Of course, we're building a model of site of metabolism, so that translates into, at least for P450s, many thousands of sites that we're training on. But yes, they are very carefully curated sets. But yeah, typically we're looking at hundreds of molecules as a minimum to train these sorts of molecules. And again, that's many more potential sites of metabolism that we're looking at there. One thing to mention in terms of those data sets is quality is absolutely key. We look at actually the experimental protocol used to generate the site of metabolism data for each compound. If you go into the literature, you'll find many examples where the experiments have been run at very high concentrations and unphysiologically high concentrations. That can lead to significant distortions in these sites of metabolism. And so you've really got to drill down very carefully indeed. And unfortunately, some of the published data sets have a lot of errors and a lot of examples of inappropriate data in there. So something we spend an enormous amount of time looking at.
Are the QSAR models for rate of metabolism part of our standard modeling suite to start with?
Right. And I guess this comes back to the point that I raised about building global models of the rate of metabolism. So here, in our experience and in others, really because of the complexity of the catalytic cycle and the many different factors that go into determining the rate of metabolism, it is not possible to build a global model of rate of metabolism that is sufficiently quantitative for us to produce it as a product, to be quite honest. I know others have, and I've seen the published statistics, and I'm afraid I wouldn't include them in our product. So there is not a single model that is part of our software to predict rate of metabolism. However, as I mentioned, it is certainly possible to build local models of rate of metabolism. So combining the metabolism module in StarDrop with, as I demonstrated in this example, the auto modeler to build local models, then yes, certainly you can build rate models of your own data, really quite successfully.
That moves us on to the next question quite nicely, because someone's asked for a little bit more information about this specific model.
Yes, indeed. So reading the question myself, Catherine, sorry, I jumped ahead. So you mentioned that building model 100 structures seems like quite a low number. And yes, I certainly, if you were trying to build a global model of rate or of metabolism, you would, that's a trivial number. The difference here is twofold. One, because this composite site lability is a very mechanistic descriptor, it's not a simple structural descriptor, it's actually capturing information about the activation energy of each individual site on each molecule. That contains a lot more information than your typical sort of cheminformatics type descriptors. And what that means is we can build models on much lower numbers of compounds. But come back to my previous point, that's not true for a global model. And so, yes, one of the reasons that a small number of compounds could be used in this case is that it was a local model of a single project's chemistry. There were a few different series in here, but a single project's chemistry, and a relatively small number of factors were responsible for determining the rate. Primarily composite site lability, but also binding affinity that is really governed by, or not governed by, but influenced strongly by the log P and the pKa. And that enabled us to build this very, very successful model in this project. I should say that it wasn't tested on 13,000. We did actually have a separate independent test set that we used to validate the model. We then applied it to the 13,000 compounds, which was actually used in the project to select these 40 compounds prospectively to identify these stable and potent compound series.
Great, thanks very much. I think we've got time for one last question. So our final question of the day, can metabolism be predicted for blood plasma peptidases and other than liver enzymes?
Yeah, another good question. Yeah, always keep us on our toes, our audience, don't they? So in principle, yes. Other enzymes are amenable to this approach that we use, this accessibility and reactivity approach that I mentioned here. It's a very general approach to predicting metabolism. The challenge here is data. If we have good data, it's a research project. It would not be trivial, but yes, certainly we were very interested in predicting metabolism by other classes of enzyme, including peptidases, and with sufficient data to train the model, then yes, we're very interested in doing that. And of course, we are looking at other enzyme families at the moment, and it is research, so there's nothing guaranteed, but we're very hopeful that we'll be able to add to this portfolio of different enzymes in our metabolism predictions.
That's great. Thanks very much, Matt. I think we will leave it there. So all that's left to say is that this webinar recording will be available on our website later, and get in touch if you've got any other follow-up questions. And thank you very much for watching.
Thank you all very much.